Okay, today I'm going to talk about the fourth of our biomolecules, which is nucleic acids. So we've talked about proteins, we've talked about carbohydrates, we've talked about lipids. Now we're into nucleic acids. So nucleic acids literally put the Na in DNA and RNA, and they really have two major functions in our body. The first is the home of our genetic information, which really comes from the DNA side of things. And they also code for proteins, which really comes from the RNA side of things. And DNA and RNA stand for deoxyribonucleic acid. So there's kind of the deoxyribonucleic acid. So we have our D, here's our N, and our A. And then ribo, this ribo comes into the other one as well. We have ribonucleic acid, RNA. Okay. So ribo refers to ribose, and we'll talk about the structure here in a second. Just like the other biomolecules, nucleic acids are made up of smaller individual repeating units, and the monomer, or the individual repeating unit for nucleic acids, are called nucleotides. Okay, nucleotide, that's the individual repeating unit that makes up both DNA and RNA. And a string of nucleotides gives you a gene. So when you've talked about genes and genetics, you know that there are specific things in your DNA that code for specific traits. So if you have blue eyes, for instance, or if you have dark hair, or if you have varying features, freckles, all those kinds of things are controlled by these sequences of nucleotides. And we're not going to get into the details of that, but just so that you know how this relates to the genetic piece of it, a string of these nucleotides that codes for a specific characteristic is called a gene. Now these nucleotides look a little bit like this. They're made up of three distinct features. One is ribose, and I didn't draw everything coming off of it. This is really just a stylized nucleotide in general. So this whole thing is a nucleotide. And at the heart of it here is the ribose, and if you have been looking into carbohydrates, you know that ribose, the OSE ending, indicates that it's a sugar, and it's just one individual ring, which indicates it's a monosaccharide. So ribose is one of our monosaccharides, one of the individual repeating units that makes up carbohydrates. So ribose here is, and again, it would have multiple alcohol groups hanging off of it and all that good stuff, but I didn't want to draw it here for sake of brevity. It's attached to, on the outside here, a phosphate group. Phosphate is a polyatomic ion. It comes from usually phosphoric acid, some sort of reaction with phosphoric acid, but PO4 to the 3- is our phosphate group that's out here. And then we're attached on the other side to some sort of what is called nitrogenous base. So just from the name, you already know that nitrogenous bases probably contain the element nitrogen, and you would be correct about that assumption. So nitrogenous bases, and I just have a generalized, this looks like a cyclohexane or something, but I just have a generalized structure here. This could be any one of the nitrogenous bases in either DNA or RNA. So for DNA, our deoxyribonucleic acid, there's 4 different nitrogenous bases, and we usually just abbreviate them A, T, C, and G. A is adenine, T is thymine, C is cytosine, and G is guanine. And each of these bases has a unique structure, but there's 4 different options, and they all contain nitrogen. So just kind of a basic overview of what these nucleotides contain. They have this phosphate group, it's attached to our sugar in the center, and this nitrogenous base. That makes up one individual repeating unit of our nucleic acids here. So that's for DNA. RNA has the same bases except for one. Instead of thymine, instead of the T base in our DNA, that's this one here, we have U, so that would be uracil. So we have uracil instead of thymine in RNA, and that's the difference between the two. And so there are specific ways that these nitrogenous bases will base pair together. So adenine pairs with thymine all the time. Cytosine pairs with guanine. Okay, and if we're talking about RNA, then because there is no thymine, adenine pairs with uracil. So if we're talking about RNA, that would be our U instead of a T. And these are paired together. The way that they're attracted to each other is because of hydrogen bonding between the two pairs. Okay, and we'll talk about a little more detail about that, but comes back to hydrogen bonding, these intermolecular forces, these dipole-dipole attractions to each other. Now, just to give you some perspective here, in one molecule of DNA, there are around 247 million base pairs. This is one molecule of DNA. Okay, so 247 million base pairs. And in one cell, so one cell in your body, you have a nucleus, right? There's a nucleus at the center. The nucleus contains all of your genetic information. And in that one nucleus, it contains about 6 feet of DNA. So if I was to stretch out your DNA molecules, your DNA molecule that's in your nucleus there, and stretch it out along a meter stick, I'd need 2 of them, right? You'd have about as much DNA in one nucleus, in one cell, as you are tall. Okay, so that's kind of hard to visualize, that something so tiny can take up such a large amount of space when stretched out, but that's how compact it all is in there, and that's how tiny these things are, if that gives you another sense of scale. Now, in terms of structure, I said that ribose was a major component. This is ribose. Again, it looks like our monosaccharides, so we have our hydroxy groups, our alcohols that are sticking off of it in various areas, and it's a 5-membered ring with an oxygen, so 1, 2, 3, 4, 5 in our ring, and then we have our, again, alcohol group sticking off of it. This is ribose. For deoxyribose, the deoxy part, D usually means to remove something, right? To detoxify your body, for instance, if you were doing a cleanse or something. If you were going to detox, then you'd be removing toxins. So deoxy is the removal of oxygen, and it's from this position in particular. So if this was just a hydrogen instead of an oxygen with a hydrogen, so a hydrogen instead of that alcohol group, so if that was a hydrogen instead of that hydroxyl group, then it would be deoxyribose. That's the only difference between the two, is that one oxygen in that one location on this sugar backbone. And this sugar backbone has to do some chemistry with these other things in order to have the phosphate group sticking off of one side and the nitrogenous base on the other side. But this is just ribose kind of prior to doing some bonding and some chemistry with those things. Okay. Now if we build up our sequence a little bit more, then now we have kind of a sugar phosphate, what is called a sugar phosphate backbone. So we have a backbone along this side. Here's my nitrogenous base, nitrogenous base, nitrogenous base. So this part right here is called the sugar phosphate backbone. And you can see that my other strand of DNA, so here's one strand of nucleotides, right? And they're all kind of going in one direction, this direction. This other one you can see is kind of going in the opposite direction. It looks like it's upside down, so it looks like it's going this way. So these pairs of DNA will link up kind of going in opposite directions, and the details of that aren't super important at our level, but just so that you know, there's a sugar phosphate backbone here as well. The sugar and the phosphate backbone really provide structure to a DNA molecule, so it's what helps hold it together. And these nitrogenous bases here at the middle are really the ones that are doing all the genetic coding, the sequence of them. The thing that makes them unique are these base pairs here at the middle. So these base pairs are held together, again, with hydrogen bonding. So if this was A and this was T, for instance, that could be a thing. If this was C, then this would be G. If this was A, then this would be T, all the way up, right? So however that would work together. I'm not drawing these to scale. This is just for comparison's sake. So we're just kind of using them as a way to illustrate what this looks like. Now these strings of nucleotides then are held together with hydrogen bonding at the middle, and they pair in a specific way that we can predict, a specific predictable way, and then the whole molecule twists and turns. So if it kind of did this motion right here, which I tried to show with kind of a swirly, if we did this swirly business with this molecule, so if I take this molecule and I turn it and I turn it and I turn it, then you'd have kind of this ladder, and then you twist the ladder, so it twists and it twists and it twists. Then that gets you at the structure of DNA. Okay, so that's a little bit of a weird way of thinking about it. You can see some better pictures in your book. But we call that structure then a double helix. And it's kind of this idea that this ladder, when we do this motion to it, would be twisted. So the double helix is kind of a twisted ladder sort of look, and it is discovered, the way that these things fit together and the overall structure of DNA molecules was discovered by James Watson and Francis Crick. So Watson and Crick are pretty synonymous with DNA structure these days. They weren't the ones who first isolated it, though. DNA was isolated much earlier on by a guy named Friedrich Miescher. So before they even knew what it looked like, Friedrich Miescher was out there actually in the battlefields collecting band-aids, because he was studying pus, of all things, which is sort of disgusting, but that's science back in the day. And he was collecting all these used band-aids so that he can look at the genetic material. And he was looking in specifically the white blood cells. And these white blood cells are the ones that are kind of the cavalry that comes in for immune responses. So again, pus. And he called the stuff that he was studying, the stuff that he first isolated, he called it nuclein. And he actually thought that it was a type of a protein back then, which kind of reflects that this is one way that we name proteins, and he called it nuclein. And then nucleic acid came about as they were determining structure and as they got a little more information about what they were looking at. Okay, so it wasn't Watson and Crick that discovered DNA itself. That was done much earlier. But the structure of the DNA molecule was Watson and Crick, and that's why they were important. Okay, so that kind of wraps up that little section. If you have questions, as always, please don't hesitate to ask.